Hello and welcome to the Out of the Lab podcast, where we discuss the operational challenges in cell and gene therapy. Each episode, we sit down with a different voice in the space, from executives and scientists to patients and advocates, to figure out exactly where the industry is breaking down and how to fix it. This episode is brought to you by Method Made. Accelerate your tech transfer by up to 6 months using video SOPs to capture technical nuance. Take the complete interactive product tour at methodmade.ca. Last thing before we get started, quick shout out to the executive producer, Tatiana Matviva, for her work in guest selection and topic choice. Today, I'm joined with David Dismuke, the CTO of Forge Biologics. David, welcome to the show.
Happy to be here, Nicholas. Thanks for inviting me.
Great. Well, David, something we've discussed extensively on this show is a lot of these sort of operational challenges in manufacturing and the transition from R&D to process development and finally through manufacturing. So a great place we could sort of start would be, given your experience, what are some of the common operational mistakes, even before selecting a CDMO, you see in drug development?
Yeah, it's tough. I've been on both sides of it. So I've been in the CDMO world for a number of years, both in academic CDMO and Forge, large corporate CDMO, and then I've been at therapeutic companies in the past. And you're working on the therapies, and you're trying to get them to the patients as quickly as possible. And there's what the biggest issue is time, right? So how do you how do you get the design? How do you do your lead candidate selection? How do you get that right? As much in the early stages that will help you in the later stages. And it's tough, right? Because you haven't made the drug at scale. You may not know some of the things, and there's always technological improvements along the way, because it takes years to do the early work, and the field has been moving so quickly, and improvements, really even in the past 5 years, it's gone so quickly. So if you're doing your lead candidate selection, and then a year later, hey, this is new improvement, do you implement it, or do you just keep going? And so, and it could change some of the nature of your drug, and you just don't know. So that's a bit of a challenge. But I think some of where I would say biggest challenges are in sometimes going a little too quickly without knowing how, especially from the critical quality attributes of the drug. You usually don't define them too early. You may not know what they really do. And in AAV gene therapy, for example, I've only been at a few companies where for one of the biggest things we look at is empty full ratio, right? And so how many of your capsids have the gene of interest? How many don't? What is the impact of that, right? And so early on, a lot of the studies were, especially if you're doing IV administration, the impact could be very minimal. But one negative of that is you have higher host cell DNA, and so because empty particles tend to package more host cell DNA, and so you have this trade-off in how do you improve that ratio, as high as you need it, and do the studies. So we this couple of companies, we actually did studies to define what is the full particle ratio that's necessary. And it may look fine, in a mouse model or even a primate model, but then when you're at the end of the day, when you're like, well, my host cell DNA is higher because I set my ratio at 50% full particle, then you're having a regulatory challenge that you have to go back and try and solve. So I say it's hard, right? But knowing those CQAs as much as possible, knowing what you really need to do is both, is what is that effect in your model, and then what is it going to look like, from a regulatory perspective, as you think about your commercial drug. And that the other side is, I think a lot of companies move really, or a lot of therapeutics move very quickly because they want to get in and make sure that the drug works before they're able to get more capital to move it forward, or before they invest too much into that program because maybe it doesn't work. And so, but the other side of that is it needs to become a commercial drug. And what does that process need to look like for it to be a commercial drug? So you're always trying to fight these two different objectives, and oftentimes I think it goes a little bit faster than it should, and there needs to be more time to slow down and determine, is this working as intended? What's the long-term goal? So there's sort of pause points, stage gates along the way to make sure that you're meeting the objectives, not for just the short term, but also the long term.
Yeah, I mean, that makes a lot of sense. And there are so many interests and incentives at play. And when you're a venture-backed company with limited runway, you have to meet goals by a certain point. So that's a reality. Something that's come up on sort of previous episodes as well is the risks when you do move too fast of having to go back to the R&D bench once you're coming through that GMP area.
Unfortunately, I've seen that far too many times, right? And so either if you don't do it, then you run out of capital, right? That's the problem. Or that program there's a lot of good programs that die on the vine, unfortunately, because in the long history, of course, in cell and gene therapy, where it was the wrong capsid, wrong dose, and the therapeutic gene made a lot of sense. And of course, early days, everything was AAV2, AAV1. Now we have the novels that I think are pushing the field forward, but the technology on one aspect wasn't where it needed to be, and it was the death nail for that program. But the idea was sound, the overall technology was sound, but yeah, it's understanding, does this work before you invest too much in it, and then being able to go back, I think is important. Probably another thing I would always say, I give this advice a lot, is don't stop innovating. And so there's a tendency, once you have a lead candidate, once you have this, is okay, we're done.
Yep.
And there needs to, I think, a little bit, not a lot, but a little bit of still investing in some of the as what you want to call it, your next generation drug, or as a backup. But to have that, because if you do have to go back, you've saved potentially a year or more in years potentially, and because again, the ideas are usually very good, and one aspect can sink you. So.
Yeah, that makes a lot of sense. Now, it sounds like you're sort of advocating for a product perspective when we're talking about critical quality attributes. Yeah. One thing I've sort of noticed is, like, obviously research happens in a very scientific way. It's like we're thinking as scientists, but then as we're moving towards manufacturing, it becomes more of a engineering discipline. Now, for an audience who's, a lot of them are early stage preclinical asset developers, some of them on the sort of technical side, how would you advise sort of thinking about, as you're still, it hasn't fully formed, but you're still doing the science, how can you think about these CQAs in such a way that it'll go relatively well when you're actually approaching the tech transfer time?
Yeah, it comes down to analytics. And, it's an area that, has also improved quite a bit over the last few years, whether it's, more mass spec analysis, obviously next gen sequencing, really understanding your drug as much as you possibly can. So, so investing in, and sometimes it's, it's not really a huge cost. It's just making sure that you look at every critical quality attribute that you can, even if you feel like it may not be as critical. There's a lot of, especially in the US for the FDA and other markets, it's a lot of report result, sort of trending that, understanding, we tend, and I've seen it for the last 20 years, you tend to focus on titer. Did I get the titer that you need?
Yep. Yield.
And yield is king there, but the other attributes are really important, and they may be more important than you realize early on. And so, and there's, again, there are better tools for this these days. Empty full is always. I'm old enough to remember when the empty full analysis that we did was, I'd go in the lab and do transmission EM and then circle the empties and fulls, whether they were dark circle or or light. And so, terrible analytic, but that's what we had at the time. It's what we employed. And now you have, CD mass spec, you have, the refined system that's, mass photometry. You're allowed to, able to do really small amount. Cryo-EM and AUC are still the gold standard, but you're having better tools to do that. It sometimes doesn't cost you that much. It doesn't cost that much vector for mass photometry, and you get really good results out of it. And so I think really paying attention to those things that maybe you think are less critical because you're focused on yield. Do we get enough yield? And then always being kind of up to date with what's new in analytics. So analytics sort of rules.
Yeah, well, something I'd like to kind of get into in that area is in cell therapy specifically. It seems like there's a unique value in analytics in that we've got such a heterogeneous group of patients. And so on some of the previous episodes, we've talked about different predispositions and diseases that patients might have. And it seems like sometimes you need that flexibility and that data to make decisions when you now have a patient who isn't representative of all of your previous testing. So maybe we could talk a bit about the implications there.
Yeah, on the patient side, sorry, make sure I'm getting the question.
Yeah, I mean, as a drug developer, you're developing a drug, and you are, whether you're engaging in a clinical trial, progressing, however you're doing that, you're starting to get some patient samples that may not be as similar as you might have initially expected to the rest. And I wonder how having robust analytics can help you in those types of situations.
Yeah, I think probably the thing I focus there when you're mentioning that is patient selection, and so knowing your patient population and pharmacogenetics and how that may be imparting what you're reading out on your clinical trial. I've seen obviously a lot of that. The other part of that is just your dosing. What's your route of administration and your dosing, and so. It obviously gets very, very complicated, right? We tend to think of NABs and TAB assays for that. So on the analytics side, certainly develop my share, and that's, I think, across the field, there's really not enough consistency in those assays, and it varies over time. You take the same patient and you test them. Their total antibody or neutralizing antibody may be very different, depending on when you look at them. So again, analytics is important, but it's not a single time point, right?
Yeah.
That's probably what I would focus on there. But understanding, in the really the broad spectrum, how your quality, your vector analytics, but also because again, your empty full or your partial particles, your route of administration is going to change how useful a partial is, right? If you're doing an eye trial or a really focused area, partials will able to transcomplement one another, and you'll have more functional outcome. So you can maybe use have more partials in your prep. But if you're doing IV and your targets are really, then you're not going to get that sort of a transcomplementation, and they're not going to be effective. So it's holistic, I would say, and really thinking about those aspects. So I don't know if I answered your question, but you can't look at a single point. You have to look at everything and what you're trying to do. And I think that's where, is a sort of aside, cell and gene therapy still relatively new, and but it's changed a lot in the past 20 years that I've been in it, where so much more expertise is available And so, than it used to be. So now you have, so I think people are looking at it more holistically, and it's not just a few experts. There's a lot of people who know how to look at things appropriately and make informed decisions. So,
that makes a lot of sense. And it's really different when we're talking about these new fields because there's a lot more unknown unknowns that are affecting this whole process. There's things happening in these tanks that we still don't know exactly what they are. And one thing I'm sort of curious about, thinking about the analytics aspect, is given that there are these many blind spots still out there, does it give you a level of sort of flexibility as new data comes to light, if you have a lot of analytics, to make better decisions throughout your clinical development process?
Yeah, I mean, I don't think it's ever too late to learn from new information, right? So, even if you already have a commercial drug, there's post-approval changes you can make that are in every AV manufacturer has done them. So I don't think that's unusual. We shouldn't be shy from it. As better information comes in, we need to employ that into our decision making, and to make a more effective drug. To make the drugs more vector per batch, right? So we need to be doing continually improving, and not just be a line in the sand. Okay, we've crossed it and we're done. And as new information, put blinders on. We're not going to look at that. So,
yeah, that makes sense. Now, it sounds like you really advocate for this sort of continuous improvement model. And one of the sort of challenges, the structural challenges I'm seeing, is it's really easy to iterate quickly and do things like high throughput screening when you're in R&D. But once you're moving into the GMP place where there's heavy regulations and documentation, and things, just because of the way that they're structured, move slowly, it becomes really difficult to experiment at that phase. So how do you kind of manage those types of situations?
Yeah, it's a great question. I think it needs to be structured, right? You get into GMP, you can't be making changes on the fly. So you need the structure, but the structure that really sits there, and to make sure that you're making those informed decisions, goes back to analytics, and really in the AAV world, in viral vector world, it's around your potency assay, probably more than anything else. There's less to hang your hat on, quite honestly, from an analytics side, that really tells you, did the process alter the vector. And so I think if you had to invest in one thing more than the other in early stages, it's really coming up with a good sound, low CV potency assay that you can ideally run at high throughput. So you can make these changes, and and then assess, did it impact the functional outcome of the vector? Because generally you don't make a lot of changes that change the purity profile. Quite honestly, if you look at your capsid purity or your transgene purity, there unless you make changes to the design of the transgene, you're usually not going to make that many really big changes in the packaged content. Process changes you can make, but again, that should read out in your potency assay. So if you have a lot of partials, you should hopefully see that in a good potency assay. And where I think it's a challenge, because especially from a good functional potency assay, that can take quite a while and can take a bit of work, but I think it pays dividends in the end, to really know whether it really changes your outcome of your vector when you make those changes. And it also helps you later on, because things are going to come. Improvements are, the field is not static. So improvements are going to be developed in areas you don't know. And when they're applied, you'd say, I'd love to be able to get 2-fold, 3-fold yield out of my bioreactor. So having a good way to put in a comparability study, which is going to, really hinge oftentimes on that potency assay. If you don't have it, then it becomes a stumbling block instead of an advantage.
Yeah. So really planning for improvements as they're coming down the line at the beginning.
Yes.
Yeah. Now, one thing I'm curious about with these assays is whether it makes more sense to do these sort of at the R&D level, whether it's an academic or CRO, or does it make more sense once you've sort of transferred it in say, the open process development lab at the CDMO that's actually going to be doing the manufacturing? Does it make much of a difference?
Yeah, I mean, Forge, we have a really, great process development, analytical development teams, and I think that's, so a lot of clients utilize our capabilities, but then we also have clients that have their own internal, PD labs, R&D labs, and so I think both models are great. And I've been at companies where we did all of our PD work internally, and then we tech transferred it to the CDMO. So if I had to pick for myself, I would build my own. I like having my PD lab, so my AD lab, so I would build that myself, and really have control of the technology. Probably again, focusing on analytics would be to have in it. Having the analytical team is probably, if I had to choose one, would choose the analytics side, and then then the PD. But I do think it's an advantage if you have it internally, so that you're not as reliant on the CDMO to especially Forge, we're busy, so we can't always just go run it the next day. If you have your own lab, yeah, you can do that. So
yeah, that makes a lot of sense. Now, as we're sort of thinking about companies that are preclinical and this might be their first time going through the drug development process, it'd be interesting to talk a bit about the sort of development strategy and why manufacturing shouldn't just be a handoff, throwing the hat over the wall to the CDMO and hoping that it sort of lands. How can these companies really think about this while they're still doing science? To de-risk it, in a sense.
Yeah, so I do, as I was mentioning earlier about having AD or PD capabilities. I think it's knowing your drug. So it's they're still bespoke. No two AEVs are the same. We have about 60 clients at Forge, and I promise you, they're not the same. And some of the challenges that we have in one vector are not the challenges we have in another, whether that may be an aggregation or some higher residual. And it goes back to, that vector and how it's produced or something unique about it. There's some commonalities, but a lot of it, when you get down to where you're going to run into roadblocks, are the things that are really product specific. And so expertise, whether you hire that expertise and you have it inside your shop, and then you're able to utilize that with your CDMO. You can't just throw it over, or you have really good consultants. And again, like I was saying before, the field has gotten much better, and so there's a lot of expertise, and there's some great consultants. We work with companies that are essentially virtual. They have no lab space. They may have a really small office. But they have expertise in-house, whether that's their own, small team, CMC expert, or whether they have used one of these third parties. So I think it's the expertise is out there, and just make sure you have that inside your own company, so that you can, again, whether it's a consultant or FTE, so that when you run into challenges, you have somebody on your side who's looking at it and trying to figure out how do we solve this issue.
Yeah, that makes sense. So some CDMOs they offer a sort of service as well for transferring in. So what is your sort of take? Do you suggest they have their own CMC consultant to sort of cross it over the line, or do you also offer some level of support as well during that period?
Yeah, so I mean, good tech transfer, right, is a two-sided structure. So if you're really tech transfer, and so we call it tech transfer if we just received the transgene, right? If we still need to look at that and make sure that it's that we're not going to run into issues. So there's information transfer.
Mhm.
But we work with a number of companies that that have developed their own process. They tech transfer, but it's two-sided, receiving party and sending party, and they need to be working in close concert. And so in an ideal world, right, you would have everything fully baked, and there'd be no differences in the equipment, materials, background that you're working in. And that doesn't really exist unless you're building your own plant and tech transferring it somewhere else. And even then it's difficult. So I think it really comes down to, to understanding how those differences may impact, productivity or product quality. And I think, we've been, knock on wood, we've been successful there where, but you're really, really understanding the full process. And, in an ideal world, we would receive, fully written batch records and, step A, step B. That usually doesn't happen. It's here's the process. It's more of an outline than a fully defined process. And then you work on it together to see how it fits your, facility fit. But again, that really means people in plant in your PD lab working together, or your analytical lab, and ideally vice versa, where the team goes and works in their labs and gets to see the process as they intended to have it run. But and really trying to identify those gaps is it. So using the tech transfer process, there's more than one. There's a gap analysis that happens just in scoping, and then there's a gap analysis when you get to the your how this process is going to translate into to really fit your facility. So there should be several gap analysis on material, process, everything. So.
Now, one thing I'm curious about in terms of the tech transfer is, I wonder if you can sort of see, I don't know if you've necessarily measured this, but the impact on the time, because we're talking about these drug development companies, they're venture-backed, time is ticking, and they need to get through their manufacturing phase 2 and so on. So this investment in the tech transfer, including whether it's both teams visiting each other's sites, have you seen how that sort of pays off in terms of this company, the total time to get to a finished product?
Yeah, so you're not going to like my answer. It depends, right? And so the gap analysis should help inform, if you're running the same reactor, if you're running the same skid, then obviously that makes life easier. But if you're, if the CDMO is, can pull in that specific piece of equipment so they can, you can run it exactly the same way. But when you have these gaps, that's, where the issues can happen. So that's where you kind of need to focus. So it depends if it's really a like for like as much as possible, then it's typically going to go much smoother. But if there's a lot of, gaps or differences between the process, then you should invest more time there. So it's going to stretch it out, but you don't want to just skip over it and just assume it's going to be okay, because oftentimes it won't be. So, but that's where, having those teams work closely together. So
one thing that would probably be helpful for, companies ahead of this is understanding the sort of perils. So one question is like, what does it look like when it doesn't go as expected?
Yeah. So, I think for the most part, it goes well. Where I'd say it's been probably more of a challenge is because, growing cells in a bioreactor, whether you're using a Cytiva system or a thermo system or a Paul system, or it doesn't probably matter as much. Where you get into the downstream process, and especially running an AEX, for empty full enrichment, that's where I think it, that's a pretty critical step. And so I think that's where it can be a little bit more challenging if you're not having the equipment that's similar. I mean, the resins are pretty similar, but overall the design of your chrome column and the system itself, they're going to run a little differently. whether you're using a Repligen skid or using, an Acta. So understanding those differences, I think is important. I think what's challenging too, it's gotten better, is having GMP equipment. I'll give an example here. Is there, for a long time, there haven't been small scale GMP chrome skids. And so that are single use for so AAV, we're typically single use upstream and downstream. And I've seen this throughout my career where you're using multi-use equipment and then trying to do cleaning validation on that. It's essentially a PD skid that you're having in GMP. And we don't do that. It's all single use throughout, but that's a change. Now you're going from a PD system that is typically controls a little bit differently. The flow paths are a little different, how they work. They're larger bore. And so it's not an easy transfer, right? And you don't want to use this multi-product contact equipment in GMP if you can avoid it. Maybe if it's dedicated for that one product, and then you can be less challenging. But then that suite needs to be really dedicated. That piece of equipment's always dedicated for that one product, and it got to go through all your calibrations and qualifications for that one product. So I think that's just an example of where it doesn't always translate like for like, and you're going to have that difference, but it's a good difference, right? You want to have that single use capability, and not have to worry about product carryover from the previous drug to your drug.
Yeah, that makes a lot of sense, and it really sounds like simplicity is really key here, and having everything isolated is sort of required to have that simplicity and control that you need at GMP manufacturing.
Yeah, I mean, again, I'll take a step back. That's what we do, right? And it's been now ingrained in how the industry operates. Really, it's single use throughout. At the same time, I have worked in the field a long time, and where it wasn't single use, and you would always test for prior product, and you do cleaning validations. And I think the likelihood of AAV getting crossing into one product from one manufacturing to another is extremely low, but you're building that safety into your process, even though I think there's you can probably operate in a different way where you can justify that the likelihood of an adverse event is so low, astronomically low, that it can be done a different way. But I think the field as it is right now is single use to avoid those challenges.
Yeah, so single use is the current standard. That's good to know. Well, yeah, it's been a super insightful conversation today, and thank you so much for joining us.
Okay, no problem. Thank you for having me.